So let's begin with this point about overtraining. Let me just repeat a rather simple story because it's important. The story about overtraining. I hope it's okay if we keep the window a little bit open because the room has been used for a while. So machine learning, any kind of machine learning device essentially, optimizes some free parameters. Let's call that training. Then now monitoring two data sets, the training set in green and the testing set in blue, looking at some criterion for accuracy or performance. You typically see something such as training set increasing and at some point the test set being overtrained or saturating, and that's the point of overtraining. Now there are 2 points here. The first one, implicitly, the slide suggests that you can train the problem to 100%. So at some point the green one hits the 100. Is that always the case? Why not?
See, it looks the perfect. It looks the perfect.
Okay, we're not in the social part here. Social call is over. Let's do more science now. Again, science science has to do a lot with social aspects, but this is not the part that I'm open to these discussions. Actually, it's something you have to consider. But here is a different story.
We're still in neural networks, right? Or is it every machine learning method?
Good. Thank you for making that distinction. Let's assume we were in neural networks. What does it depend on? What accuracy you can get? So you have an input, you have an output, so you want to classify patterns. Training is the data set that is labeled. Can you, in a neural network for instance, SVM or similar method, can you train to 100%?
I think it depends on the hidden layers and the output units. If you have less hidden layers, hidden units, then it is necessary to hard code the training and the text data. Then it's not possible because the neural network has to abstract.
So let's just assume that we have two examples here, and in the training data, one is mapped to 1, so the vector 0, 1, 0, 0, 1, 1, 0 is mapped to 1, and the other case, the same input vector 1, 1, 0 is mapped to 0. How can that be?
Error?
Yes, it could be an error in your labeling. In the field of biology, this is a not unlikely scenario. Or, for instance, for sub-cellular localization, I showed you that actually the same protein may be in different sub-cellular localizations, or may do something different if expressed in a different organelle, tissue type. So whether that protein is in liver or in the brain, it may have slightly different actions, right? The same protein. So in that sense, this is clearly not an error. It's just that we're predicting sub-cellular localization and we're not considering tissue type. Okay? So both things could happen. So immediately you see from whatever error criterion you put in, this cannot be trained no matter what you do. Right? Now the problem is, in this particular case, it's very obvious to see, right? So you can actually check your input vectors and look at identity. But errors could be more subtle than that. It could be that you have a very, very, very long vector and there's a tiny difference. And for all the other things, this tiny difference is pointing in that direction. And for the others, not. That is not what you can establish. So you cannot, back to what we discussed on Tuesday at the end, you cannot easily compile a correlation between these vectors. If you could, you would not use machine learning. The problem is not that simple. So, okay, now let's assume the problem is a different one. let's assume that the data is such that in principle this doesn't happen. So you never have two identical vectors mapping to two different wanted outputs. David says it has to do with the hidden units. So obviously I told you if you don't have a hidden layer, you cannot solve the XOR problem. You need a hidden layer. And for the hidden layer, you need some hidden units. We discussed how we choose them, and we talked about the number of free parameters have to somehow match the samples that you have. Say we have 1,000 proteins, we have 20 input units, so that would lead us with 5 hidden units. Now, assume my 5 hidden units for my problem. They do not get 100%. I can blow up my system. So one way to blow up my system is instead of using 5, I use 50. Come in is open. When I use 50, or whatever higher number, and I can get to 100, I know that these simple errors are not in there. Right? If I blow up substantially and can't get there, again, now we're back, it could be the errors, and they may not be as simple. Or it just could be that the input is not well defined, so that 20 input units are not enough, or that your data is not enough to support the complexity of the data. Now, let's just assume that we have a situation where we have 1,000 samples. We use 20 input units. 5 hidden means 20 times 5 is 100. You want 10 times as many samples as free parameters. So with 1,000, that's maximal what you can support. But let's just assume that the training error, the training performance is getting to a point that is just above this test performance. Is that reason for concern? Should he blow it up more? Or let's just forget about the blue line. Let's assume that your training gets to 50% with your 5 hidden units. Well, let's assume that this is the best you can do with Weka. Or Weka. So it's not only neural networks, so you try a bunch of machine learning devices. No. Wait. Let's forget about that assumption. No Weka. Neural networks. My point rests upon the question here for neural networks. So you see that with 5 hidden units you cannot do it. But with 50 you can. What do you do?
Take 25. Take 25?
Back to the social issue. Fair enough. But again, the scientific issue is, the next step is that you need to use another data set to see how much of with the 25 or with 100 or whatever you have, with the 50, how much do you go into overfitting your data? Okay? Now, let's assume that for 5, although I do not reach 100, my test set has a higher value than for 25. Which one do I take? So again, I look at the blue, I look at the test set, and I see that with 5 hidden units I get a test performance of 53, and with 25 I get 55. And say my error estimates would be here in the ballpark of 1% standard error. What do I do? Which one is my solution? Oh, come on, guys.
The lazy decision is to take 5 because it's smaller. So
that's actually a very unexpected answer. But it has a lot of truth to it. Smaller, so that is some consideration you have to make. At some point somebody's going to use, if you believe you develop a method that makes a difference to people, then one question is how much time will it cost them to run it? And it might be, so neural networks are so fast not in the training phase but in the test phase, that the difference between 5 and 25 typically is not the killer. But that may be an issue. So it just depends on how often somebody repeats that. So this may be a consideration. Keep that in mind. That there for everything, I really like what you said, because it's an important consideration, the cost and the improvement, to put the improvement in context of the cost. When we, I don't know, when we buy some new tool and we're a little bit short on money, we may ask ourselves, what is this new one, this new version of the phone giving me that the other one doesn't have, and is that worth $1,000 or not? Or something like that. So this I like that. But let's get back to the assumption that this is currently not a concern. What do you publish? Yes, and now again you give a social answer. Let's stay on science, the bigger one, because this is more likely to get me into the journal. True. Higher numbers are always more impressive. Unless, be careful, unless you get into too high numbers, then you get into the trust issue again. So we had, for instance, we had an example like that where we, for sub-cellular localization, had an estimate of performance that approached 100. It was just full stop. All the samples we had were correctly classified. And in that particular case we had 50 examples or 30 examples, and we decided to just not publish that. Because nobody trusts 100. And we don't trust 100, right? So be careful when you are in the extreme end of things. So when you claim that you reach a certain performance, again, always put the background of random. And another way of background is how well would you do if you had experimental data? So in some sense, homology inference is one way of looking at that. Or find some other ways to sort of get a feel for what is the ceiling of the method. And typically 100 is not ceiling. Typically your data is the ceiling. And there may be a way in which you can estimate it. It may be tricky, but spend a day on it. But back to the same original question. You say I published 55 because it's a higher number. Again, this was estimated on the test set. Let me ask the question differently. Picking the 55, is that sober? Kosher? Is that okay?
I mean, when you compare the number with random, and it's 25 units, then it's much more, then it actually looks different. So imagine those 5 is a coin flip. Like.
That's a good idea. So say we had a two-state system. What David says, and that is a very, very important point. Thank you for making it, David. So say random were 50. Then essentially you look at the difference to random. And the difference to random, so first of all, the statement is by standard error this difference is supported. Essentially almost or just about supported. But what used to be just a 1 percentage points difference is in fact compared to the difference to random substantial. So you move, instead of moving 3 percentage points, you move 5, almost twice. Point well taken. That's something that is very, very important. But my question here is a different one. The question here is one about sober, about okay, about correctness, about cleanliness of what I'm doing. So I claim it's not okay. And the reason why this is not okay is because the test set is supposed to serve for an estimate of performance without any parameter optimization. By going into the table in your publication and saying, hence I've learned that 25 hidden units is the best approach, hence my performance accuracy is 55. That is wrong. It's absolutely wrong, because you used the test set that you used to estimate performance to optimize a parameter. In this particular case, only one parameter, number of hidden units, but that doesn't matter. That's why you need the split into train. In most machine learning devices, you need to split into train, cross-train, and test, where cross-train is just another sample of the data set. And that cross-train set, that you can use to make that decision. And then it may well be true that for the cross-train set, the 25 looks better, but for the test set it doesn't. But then stay with the estimate for the test set, although that gives you a lower number. You may point that out, but actually you shouldn't. It's just more conservative, okay? Good. So much about one aspect of machine learning, and let's get now back to the part of the lecture where we talk about sub-cellular localization. The first part about sub-cellular localization. So now I want to classify with, in this particular case, it was a neural network. Did I present the first solution? No. The neural network, I want to predict sub-cellular localization. I want to predict sub-cellular localization in, say, 3 states. So I have proteins that are in the nucleus, proteins that are essentially the nucleus here, the cytoplasm, and the extracellular space. Let's just have the simplest example. Now, most machine learning devices have a hard time to handle non-binary decisions. Okay? How do I translate this 3-state system into something that is sort of binary? Yeah?
You can have those 3 variables as all 3 binary variables. So is it inside nucleus or not, or is it inside cytoplasm or not? And the other one can be inferred then. So if both are not inside, then it has to be imaged. So 2 variables are enough.
So, so you would actually train 3 different devices. Is that what you're implying by that?
2, because you can infer the third one. So if it's not inside the cyto and it's not inside the nucleus, it has to be inside. So.
Okay. So we already, between my misunderstanding of David and David's correct understanding of David's point, we have 2 alternatives. In my misunderstanding, we have. Is it directly to that, or you have another way of doing it?
Maybe it's a little bit further.
Then just a second. So device number 1 does 2 times 1 against all, and the other one does 3 times 1 all. What is your point?
1 against all.
Well, that's what I just said.
Yes. Yeah, that's.
So that is covered.
Before you said that.
And yeah, oh. So, any other idea how we could do it? Let me go. Let me ask David. So, for your first method, how do you choose which ones you train? So you throw, essentially, for mine, the ones that I propose, or that we proposed, you throw one away. Which one? How do you decide? Alphabet by alphabet?
So when it's a perfect data set, it shouldn't be as. Then it doesn't.
So you already know. We are here in biology. There is no perfect data set. The problem, again, even not biology. Remember, you apply machine learning because you have a tough problem. You have the type of problem that is not easy to do. So typically, otherwise you just do linear statistics or something that is very, very easy to separate. Typically, for most problems, biology are typically very particularly tough, but you have not easy problem, typically. So typically you cannot make that assumptions that all the data sets, that at this point we have a stratified situation. You could massage your data set, but even if it were stratified in terms of number of classes, you still don't quite know whether that is also stratification in terms of number or ability to predict, right? Or whatever ever other criterion you have. What is the criterion you're going to put forward? How are you going to choose? So, in fact, let me tell you that one, David. Sometimes people may not find your solution because they cannot answer that, and then they say, "Okay, then let me take them all. It's easy anyway." That's a chickening away kind of solution. Do you have an idea? No. Any idea how you could do that?
Throw away the one with the less data points.
That's an absolutely fair way of doing it, and I believe it's an even rational way of doing it. And if there is a significant, in fact, let me just turn my argument around. If there were a data set that is significantly less, that may exactly be the argument for your solution. And maybe why you argue, that's why I do 2 times and infer the third one, because the third one is not really supported by a lot of data. Point taken. But now, let's just go back a step. If say David found a solution or had another reason for doing his solution of 2, and then we still want to look at 3, how do we compare them?
We can use the third data set as additional test data.
Yeah, we can just run it and try it, right? And ultimately, on this level of the problem, it's fairly easy to try them all. You have to be careful with this decision. The moment we turn it into 18 states here, that story gets much more complicated, which is what Tanya talked about. So which is our ultimate solution. So for our ultimate solution, the problem is way more complicated. But for the simple one, we can clearly do that. Now, let's just show you an example in the way we do that with a neural network. And in fact, for the neural network, we have different networks. One network uses sequence. Sequence is defined as what I talked about, the 20 input units. So amino acid composition, that's an old version. The other one uses solve an accessible part of this sequence composition, also 20 input units. This uses a combination. And then there is a second layer. A second layer is just a second set of neural network that uses the input from these 3 to predict again. So this could be L and not L, could be, for instance, extracellular, not extracellular, and then it will be cytoplasmic, not cytoplasmic, then it will be nuclear, not nuclear. Let's look at the C, not C example, cytoplasmic, not cytoplasmic. This one predicts cytoplasmic or not cytoplasmic for one particular protein. So does this, so does this. This one simply has 6 input units, which essentially are the output units from this one, and does the same thing again. Okay? It's trained on the same data set. Trained and cross-trained, they are identical. Okay, now, what's the difference between the SVM and the neural network? Let's begin with a math idea. The original problem is you want to separate out these points, and in fact, you cannot separate out the problem. In the neural network, what you do is you blow up the number of hidden units. I have to remember that. You blow up the number of hidden units, and in the example of the SVM, you blow it up in a higher dimensional space. Ultimately, it's the same thing. Because these hidden units essentially define a higher dimensional space. So ultimately, it's a similar idea. Okay? In this higher dimensional unit, you in fact have a clear way to separate it. In this particular case, there are several separations. The SVM finds the one that is sort of maximizing the difference between the 2 sets of points. So it in fact defines 2 very similar points that are from opposite ends. In this particular case, red and green, or squares and stars. It finds the squares and stars that are closest to the squares that are closest to the stars, and the stars that are closest to the squares. And it puts a line through that, and then tries to optimize the position that optimally separates these closest. Now, what does that mean in terms of its similarity to a neural network? Let's not talk about the algorithm. Let's talk about the idea. This implies 2 things about the comparison between neural network and SVMs. And you should be aware of those applying that. Anybody can say any of the 2? Number 1 is the SVM is great because you need way fewer data points. The assumption is that these are trivial here. That means you throw them away. You focus on the ones that are closest to each other, which means effectively you need way less data. In this particular example that I show here, you have 2 or 5, you can throw away 60% of your data. And the same is true here. And that is not unusual. In fact, sometimes you can throw away much more than 60% of your data. So it's great in an environment where you have very few data points. The SVM works much better when you don't have enough data. Okay, but there's a problem here.
It's highly sensitive to errors.
Exactly. Assume that this you call a star because of a mistake. then that exactly is what the SVM will zoom into. There's some error margin that you can define to throw out points. But whatever the error margin, as long as you don't hit reality with that error margin, so when you say, so points until here can be wrong, but then the real mistake is somewhere further down, at some point, no error margin definition will help you, and that is a problem. So the great advantage is you need fewer data points. The great disadvantage is the data has to be cleaner. Okay? Somehow you could sort of iterate that by massaging the data, by throwing out things that are not very clean. The great thing about the neural network simply is that you don't need any of that. It will find sort of the best solution, provided you have enough data. And enough data is not just, again, let me repeat, it's not just the number of data points, not number of samples. It's the effective number of sample points. That means if you have too many points that are too far away and too similar, then that doesn't help the network. It needs something like that too, in principle. But it can handle disagreement. Okay, back again to the problem of predictive sub-cellular localization. So you start with a protein, you have some machine learning device that does binary, and then, in fact, it adds up. Now, since in all of this, I have this assumption of the one against all, so the solution for the three-state system or three sub-cellular localizations of three different networks, not the David solution. So, in which I only take two and infer the third one. My question now becomes, and in some sense it relates to David's solution too, assume you make the first decision, the second decision, third, fourth in this particular case. Which one is your prediction? David, in your case, you would have only 3 here, so you would have one less problem, but still with 3 you have a problem. So it's the same. How do you decide? So you essentially, in this model that we proposed so far, here we would have 4 different machine learning devices, whatever they are, SVM or neural network, and each of them would have a classification of nuclear, not nuclear, and so forth, cytoplasmic, not cytoplasmic. Which is my solution? So, of course, the easy possibility is that this one says it's nuclear, and everybody else says it's not cytoplasmic, not extracellular, not mitochondria. Great, you know what to do. But this is real life. So typically that is not going to be that simple. Typically you're going to have at least 2 here that predict some preference, maybe weak, but some preference for nuclear, say, and cytoplasmic. What do you do then? How do you resolve conflicts like that? Yeah?
So, I mean, we can summarize them with an ensemble method and let them have a vote for the best one.
So that ensemble method would then essentially be, if you want something like this, right?
Exactly.
Really? Well, this is nuclear, not nuclear. You get into the same problem. What's the output of that ensemble method? 18 states, or 3 in this, or 4 in this particular case, or 3 in this case that I have on the whiteboard. If your answer, think about your answer, and everybody else thinks about their answer to the question, I just want to make it a little bit more complicated for you. If your answer will be yes to my question, so essentially for a neural network it would be very easy for 3 states to put them out there. But then my question to you immediately, if you insisted on a yes to that question, then I would ask you, why didn't you do that in the first place? Answers. You can do that is the answer, but you have to establish that it really works. So in this particular case, we established that for the neural network, and one particular tricky way in which we tried the SVM to have several output units implicitly, both of those were not as good as the binary state. That brings us back to the same problem. my question that I had before. How do I resolve conflicts? Oops. How do I, when I have two positive ones, this one says it's nuclear, this one says cytoplasmic, how do I resolve that conflict? Come on, give me at least a simple answer so that I can shoot your argument down. Or the simplest one, of course. It's boring to shoot my own answers down. So the simplest one, of course, would be I take whatever result is stronger. So if the prediction were stronger for it to be nuclear than for it to be cytoplasmic, I say it's nuclear. That problem, okay, now I gave you an answer. Shoot it down. What could be the problem of that one? It could be. Do we need? Or do we need a bed? So the problem could be, in fact, in this particular case, it's true. Nuclear predictions are better than cytoplasmic predictions. They're always better. That means that, in fact, the average signal is higher. That does not necessarily mean that for this particular decision, that is right. So you somehow, in order to do that, you have to be very careful how to renormalize these values. And probabilities are not the right way of renormalizing them here. You have to somehow renormalize them after you assess something like performance. And then renormalize. And then you can do that. Okay? Now this is tedious. When you do the simple way, this can go wrong. And again, in this particular case, it went wrong. We tried that. Now, here's another way that is sort of related to that. And that, in fact, if you want, takes the problem at face value. One way of putting it, well, let me give you a different way of putting it. A different way of putting it is when you look at the biology of the problem. There are different types of transport mechanisms. One here is gated, and it comes from this book here, this figure. It's a gated traffic, and then there are non-gated ones and transmembrane traffics. In some sense, the major decision a protein has when it comes off the ribosome, so when it's becoming a protein, it's sort of its birth hour in the ribosome, comes out of the ribosome, and now the question is where to go. So it stands there and waits for somebody to pick it up. That really, in some sense, is the right image. And typically, the first, most important one is the question: am I going to be destined? Is my future to be secreted going out of the cell? Okay, and that is a sort of so-called secretory pathway. Don't worry, it means secreted. It means gets out of the cell. And there are several compartments through which you go. Almost everything that is secreted goes through something that is in a large inner membrane confined body, the endoplasmatic reticulum, another one called Golgi, and then to the extracellular space. By the way, we will talk about that later. No, we did talk about signal peptides. So the signal peptide, remember, is at the beginning of the sequence. It's very hydrophobic. We talked about the architecture. And the signal peptide is exactly what this is recognized here. Everybody who has a signal peptide is going on this path. How do we decide that you stay in the endoplasmatic reticulum? And the answer, I briefly talked about that at some other point when I talked about nuclear localization signal and endoplasmatic reticulum retention signals. They are on the other side of the protein. So in front, you're sort of drawn by the signal peptide signal. In your back, you have a signal. If you have that, you stay. That's why it's called a retention. So you're retained in the endoplasmatic reticulum, retained in the Golgi. And everything that is not retained, the default is go through. And then go through the cell, either sit in the membrane or completely go into the extracellular space. Now this gives you some idea how you could do it. You could do it in a way that mirrors this process. So you could, in fact, start with the first decision: am I going to be targeted for the secretory pathway or not? Okay? This would be, and then you go further down. And again, you have seen, in principle, the setup here from Tanya. It has many advantages. One advantage is that every single machine learning device you have here can be optimized for the input for its decision. Signal peptides, I told you, are at the beginning of the protein. So maybe you don't only use 20 amino acids that describe the protein, but you also somehow have something that zooms into the beginning and finds out is the beginning a signal peptide or not. And that may be one input unit, or it may be more than that, but it may be something that really, really helps you to make that decision. This one could have a different input. That is one point. The other point that you have to realize in this setup, whether it's good or bad, is that you will further down in the decisions here, the more you move down in this tree, the more you will in fact have a subset of the data. So this decision will be done for all the data you have. This decision here will only be done for all the ones that you know are secreted, that are targeted for the extracellular space. The proteins that are going to mitochondria will not be trained in this machine learning device. You could put them in to in fact give you background. But the solution, or the answer that what we found out, we tried it, it really did not work very well. Because by putting them in, you sort of get away from the advantage that you had to a very specialized decision. Specialized, again, you know how to pick the input, you know exactly the types of points you have, and you distinguish at this point really between the ones that are going completely outside or the ones that go to ER, Golgi, and other organelles. And there is something that is very different between them. And this decision here should be made only on those, and should not be confused with something that happens to the mitochondrial proteins. So that is the great aspect of it. The other great aspect, by the way, is you can set up your device, you give a talk about it, and then somebody says, well, listen, I have a data about chloroplast. How can you handle that in your system? So far you haven't had enough data. You just add another one, wherever that is appropriate. By the way, chloroplast, does anybody know about the biology of chloroplast? It's plant specific. So we don't have chloroplast, only plants have them. But again, so this is another beauty. It's very, very easy to extend. You can keep the same system in place and just add another decision at the end.
This can't be placed everywhere, right? This can't be placed everywhere, right?
No, this should be placed wherever you believe it makes most sense. Most sense is defined as, one way of looking at it is you follow yeah.
What about the input if it changes the input? For example, the chloroplast needs more input.
But again, so this is a decision that wherever it goes, in this particular case, I can't even remember, I put it here because I assume that we put it after the cytoplasm.
It is restricted to the input of the previous,
yes.
The upper layer. Right?
Input as in samples, yes.
Oh.
And that is essentially the same question. As long as I know where to put it in my hierarchy in a meaningful way, the moment I know where to put it in a meaningful way, that moment that decision is supported by the word meaningful. So now the argument gets circular. But the other, so this is one particular solution to dichotomy, to one particular solution to finding a tree. Essentially what we have is 18, or in this particular case here, 3 binary decisions, right? Here we have more, for log tree 2 in the end we had 18 different decisions, but there are many ways in which I could have constructed the tree. I argued that this one is supported by the biology, so that's why I take it. How else could I do it? At the end of the day, I will ask you, of course, to compare the 2 ways. The else way and this way. But can you think about any other way that you could try? Let me point out to you that this is the type of problem for which it does not take that much CPU. So you can relatively easily compare a couple of different ways. Back to your argument about time or cost. So maybe you have to write up your thesis now, and maybe you don't have the time for that. But in principle, this is not going to be the victim of CPU time. So think about another way. Or for the next student, put it into your conclusion. Everybody loves that. What I didn't do, and somebody else has to do. Nobody else ever reads that and never does it. I'm not discouraging this kind of thought, but whenever you put this kind of thought, and most theses, in particular in computer science, end with that. Just before you phrase it, before you write it, before you say it in your talk, just imagine that nobody would ever do that. How would you rephrase the sentence? I'm saying don't wipe it out, but be aware of this reality. Anyway, back to that story. So how else could you do it? Come on, you want to publish high numbers. Let me give you a clue. How can you crank out most of this system? Can you arrange it such that the probability of succeeding is higher? You essentially try them all and use the one that gives the highest value. Right? Certainly for the example here of 3, you can easily do that. For when we began doing this here, Rajesh Nair was the one who did it. Here, Rajesh. This one was not in there, this one was not on there, but this was already the system. And for that system, in fact, somebody later did a thesis on this and simply tried all possible alternatives. And found that what Rajesh had used intuitively, trying to copy the biology, was in fact the best solution. But that, in this particular case, was, in my eyes, total happenstance. Again, total luck, or whatever you call it. If anything that I do works the best, then that stinks. That cannot. We didn't design it that way. There's just. And if you believe that the biological pathway is this way, and that's why, that looks like religion to me. So, not sure. I never believe things like that. Okay. Some other methods. And
but the tree method now, it doesn't include the proteins which can be in both positions. We told about those proteins which are.
No.
So only for one position.
Yes, that is absolutely right. So in this particular case, we in setting up a method, we carefully make sure that we throw all the experimental data out that has proteins in 2 different subcellular localizations, although we know that it can be true. We try to, which again is there's a longer story. And I believe so Tanya recently collected data that suggests that we did right. In the past, we only did that because we didn't know any other way of doing it. We just looked at experimental annotations in different compartments and used them also for training, for both, and it didn't work. Okay. So that was statement number 1. Then we kept them out and hoped that one day later, we have been working on this for over 15 years now, and one day later we would solve it. And Tanya recently, in fact, tried to solve it and found out that these data points are very, very unclean. So in fact, the number of annotations you have is proportional to the error. The more annotations you have, the more likely it is an error, although we know that all the proteins at some point move around. The definition of shuttling in the cell is that they move around between compartments, right? Still, there seems to be an absolutely dominant signal for some native compartment, and that is obviously to the high proportion built into the system. Okay, some other methods, and I go back to in this particular case, p-sort is history. So p-sort, I briefly mentioned that Kenda Nakai Minoru Kanisha is the idea you use amino acid sequence alone, plus then they put that into a tree. So you can predict transmembrane helices, you can predict cleavage sites, you can at some point use the amino acid composition, and you can build a tree. Down here is amino acid composition, but there are many decisions in order to place proteins into the subcellular compartments that you can do by combining methods that are out there. And that simply is the original idea. So you look at the problem, you look at what solutions there are, and you essentially build a tree on a piece of paper and put that into a software. Okay? p-sort 2 is the same thing, but now we sort of use statistics. We use sequence signal, the k nearest neighbor. So we combine that with something that really is a consecutive stretch of something that goes on through the protein. So this essentially, many of these motifs that you see here are going back to what I talked about prosite. So those are relatively short motifs. K nearest neighbor is a relatively short motif. You find things in prosite, you find things in the database, you find signals, you find methods, you combine all of that, and that is sort of giving p-sort 2, and is done by Paul Horton, and is undoubtedly the most used program to predict subcellular localization in the world. Wolf p-sort is the same thing, more, if you want to call it modernized. This is 4 years old, and is available publicly. An important extension of other p-sort methods. But again, the most important idea of p-sort is you use a couple of relatively simple methods. You build an architecture of the hierarchy of the decision making, and essentially you find whatever decision you can make according to that hierarchy and put things in. It does not train, just a sec, it does not train on one particular part of the decision. So the problems that I had before are not addressed here. Yeah?
So what if on top of the tree you make a mistake? It's kind of affect the whole result.
You mean, but that is more valid in the system. Well, it is actually in both. Yes, you're right. So in principle, this is a tree. What we had before is a tree, and I will get back to that issue. When you have a mistake, it's difficult to recover from the mistake. So let me go back in my slides. And in some sense, this is more relevant here. Say this classification you believe is a secreted protein, and it's not. So all it will see here are signals that are all wrong. Because it will see all things that are wrong, at the end of the day, some decision here will be very weak. Right? No. You don't know about that. You can absolutely not tell. It could be that the wrong signal here gives you a very strong signal, and for instance, it's not that. Okay? It could be that one of these networks, or one of the SVMs, or whatever it is, says, this is absolutely not mitochondrial. And boom, very strong signal. Which in fact is comparable with the fact this was wrong, it is this one, which means it's not mitochondrial, but you would not learn anything from that necessarily. And sometimes it may just be that it very strongly says it's not mitochondrial because it never has seen the data. Or there could be all kinds of non-good reasons why the behavior here is not the one that you get from typical proteins. So from seeing the signal here, you cannot infer whether it's there. What could you do to recover from mistakes like that?
Don't decide between 0 and 1, make it more like a probability.
Yeah, then we get, you're totally right. So then, you mean the probability down here?
Every decision, so
Yeah, but that is the problem, that every decision has a different background probability. So that's what I talked about before. It's very, very complicated to normalize.
okay.
Because, in fact, in some sense, this biological solution, as I may call it, has worked best because this was, in fact, the safest decision to make. The first one was the decision for which you have the best information. And that means that the signal that you get for the probability here is very different from the signal down here. The problem gets increasingly difficult. So there's a trivially built-in fact that if you want to get for the safest part, you move up in the hierarchy, which is actually not the right solution. So no, that doesn't get us out. Any other idea?
But this is the best way in the end, right?
Yeah, but again, so we have a problem. How do we solve that problem? The problem is that we make a mistake very early on in my decision process, and I do not have a way so far to recover from that decision or from that mistake.
Maybe just for the tougher methods, try both. Like,
Yeah, well, but again, no matter how you normalize the probability, the decision was not here, so it was in that direction. It was a mistake. What else can you do? So you could take out the hierarchy and try a different hierarchy. But since that is the best hierarchy, most of the time that would actually go wrong. In some cases it would give you then there would be a hierarchy that gives you the right answer. But on average, since that is the best, the definition of the word best is most likely this will not work. It will make it worse. So what else could we do? Anything? I don't really know what else we could do. So that is, in fact, a problem that we are stuck with. And that problem is I showed it on our own method, but in principle it's the same thing here. So no matter what type of tree you get, in this particular case even worse, because in this particular type of p-sort here, the type of decision you make here, or the type of device you have for that decision, is a very, very different device. In our case, we use machine learning device. In this particular, the last solution there for the first version of log3 was all SVMs. They use different input, but in principle it's sort of the same way of doing it, while these are totally different methods. And comparing probabilities for them is even less easy. Okay, Wolf p-sort, other methods. And that is where we're getting back to Sherlock. Let's talk about Sherlock. So I showed you at some point that keywords, so the functional keywords in a protein in Swissprot, can help you very much. You can use them to predict sub-cellular localization. Now, the idea behind Sherlock is, what if I extended that concept and went much more, I can't remember who, when we discussed the keywords, talked about just doing free text. There were 2 or 3 people in the room with this idea of just using free text analysis, building a dictionary. You were one, I forgot the third. And here's essentially the same idea. Use abstracts, map these abstracts in simple text mining to sub-cellular localization, and use that and combine it with methods such as the one that I showed you before. So something that uses amino acid composition, p-sort, or whatever method you use, sort of put that on top. Multilog is in fact one of those methods, much more advanced than ours. Our originally was, so this is a query sequence that goes through a bunch of decision making. So here's the motif search, and this idea is really going back to p-sort, where, for instance, here is this one nuclear localization signal motif that I showed you before. That happens to be a not very well working ER retention motif. Then you have a bunch of methods here that predict particular compartments with particular input. You put all of these predictions here together in a set of all against one, or one against all type of methods, and train an SVM. Principle the same thing that I had before, with just more detailed information as input. Cello is in principle using a similar idea as p-sort 2. Is this K segment, so you're looking at segments of a certain length, L, that match in some other proteins for which you know the sub-cellular localization. So you could call that homology-based inference. Before I showed it as K nearest neighbor. For p-sort 2 that introduced it, Paul Horton called it K nearest neighbor. Here, in cello, it's the same thing, but for cello, actually, the trick is that the motifs can have gaps, and the motifs in the particular way they code for the input here. They have a sliding window that they run through the protein, but the particular way is that the residue, the amino acid is not easily an amino acid, so they group the amino acids in a way that allows them to do this in a very efficient way. There are more detailed predictions, and that in fact at the end of the day, ultimately you could hope that you can recover from mistakes you did before, because you will come at some point where the detailed prediction of positive subnuclear will actually tell you, right, we are in the right place, because there will be something else that will map to it, and that is in fact the subnuclear prediction, and the subnuclear prediction was in fact pioneered by Michael Burden, who used Bayesian networks. And that he will present, I'm not entirely sure about his jet lag. So Michael Burden is also a co-developer of the automatic motif-based methods, MIME, for instance. And Michael will present this next week. I'm not entirely sure whether that will be, he comes from Queensland University. and will be here next week. Not sure whether it will be on Tuesday or Thursday. So there are, I believe there are a couple of other methods that I suppressed here. In fact, there are many other methods. But at the end of the day, I believe that the method that Tanya developed really absolutely stands out in terms of performance and possibilities it has. So there are various aspects why it performs better.